morning. My name is Terica Curtis. I am a senior biology major at Alabama A&M University. My summer project was the improvement of sprouting of turmeric rhizomes using atmospheric pressure jet plasma and plasma activated water. My host institution was Alabama A&M University, and my mentors were Dr. Rob Mintretti, Mr. Lam Dong, and Ms. Trang Pham. So starting off with the introduction into the world of agriculture and plasma, we have to address the issue of the demand for food production. We have a ever-growing population due to a lot of advances in medicine and science, and so that increases the demand for food production. In the early 20th century, nitrogen fertilizers were invented, and that actually helped the increase of food production. But to those nitrogen fertilizers actually contribute to environmental concerns such as greenhouse gases and runoff waste. So we're looking for an alternative, and they look to low temperature plasma as an alternative because it uses less energy and it also produces less waste. This was actually observed when they used plasma treated water to treat rhizomes and seeds, and they saw that there was accelerated sprouting and germination. And so they wanted to see how the direct plasma jet would work, and they saw that there was also accelerated sprouting and germination, but there was also benefits such as microbial protection and improved nitrogen fixation. It is a great alternative, but however, there are specific optimal conditions that are needed to avoid the damage of seed and rhizome DNA. So this project explores and observes those conditions and the improvement of early sprouting and plant growth of the important medicinal crop of turmeric. So our objectives, the duration of this project were to determine which of the treatments of atmospheric pressure plasma jet, plasma activated water, both or neither would be more efficient in the acceleration of turmeric rhizome sprouting, and also to discern the effect of atmospheric pressure plasma jet on top and bottom portions and whole turmeric rhizomes. In experiment 1, it was the treatment of turmeric rhizomes with atmospheric pressure plasma jet and plasma activated water. So we used a variety of turmeric that is dubbed as CL3, and we used direct exposure of the rhizomes and water to direct plasma jet for 90 seconds. And then we immersed the appropriate rhizomes in the plasma treated water for a total of 30 minutes. Our treatments were a control, which there was no treatment of anything. The second was the treatment of direct plasma for 90 seconds only. The third treatment was the treatment of the rhizomes for 90 seconds and also the submersion of plasma treated water. Our fourth was the untreated rhizome, and then the submergence in the 90-second treated water. And then we have the fifth condition, which was to submerge an untreated rhizome into untreated water. So in figure A and B, which are on the top left, you'll see the autoclave that we use to sterilize our containers. You'll see figure D, C, D, and E on the bottom left. Those were the collection of deionized water. In figures F, G, and H over to the side, we have the plasma lab that we used, and this is the treatment of the rhizome directly and the treatment of the water directly. In figure I and J, we have our treatments after they had been treated. So treatment 1, 2, 3, 4, 5. And in figures K and L over to the right bottom, we have the treatments after they were planted into their insertion trays. Experiment 2 was the treatment of halved top and bottom portions and whole rhizomes with atmospheric pressure plasma jet. We had 3 treatments, a top half treated and a bottom half untreated, which was our control. Treatment 2 was the bottom half treated and a top half treated, untreated, which was a control. And then treatment 3, which was a treated whole and a untreated whole, which served as a control as well. Here we have in figure A to the top left, the turmeric plants that were going to be inserted into the field. They were being lined up before their insertion. Figure B is just another shot of that before they were planted. As you can see, we had 4 replications. So these 4 rows here were the rows that we worked with this summer. And this is just another shot to the right of just the rows that we used. So for our results of experiment 1, we saw that the treatment 3 was the treatment of direct plasma to the rhizome and the submersion of treated water. We saw that this actually was our fastest sprouting turmeric treatment. It was actually about 4 times faster in sprouting than it was the control, which was pretty awesome results to see. We have treatment 2, which sprouted only 2 days after the treatment 3, and that treatment 2 was actually the rhizomes that were only treated with the direct plasma. Treatment 4, which was treated with the, it was submerged in the treated water. It was untreated rhizome. It sprouted 3 days after treatment 2, which is pretty interesting to see. And then we have treatment 5 that only sprouted 2 days before the control, and that was because of the water. If you submerge rhizome into water, it would accelerate the sprouting rate. And then our control had the slowest sprouting time, which was at 15 days after treatment. Okay, so for our figure to the top left, it was figure A. This was June 12th. And at this time, treatment 3 was the only treatment that had sprouted, again, after 4 days after treatment. You can kind of see in the back row where those sprouts are coming out of the soil. On the bottom picture, figure B, we have June 25th. This is where all the treatments had begun to sprout. You can see the sprouts coming out of the soil. Figures C and D are at the top right. We have both our July 7, and these are just top and side views after the sprouting had commenced. And as you can see, they have pretty decent growth rates. And July 13th, this was also another side view of the turmeric treatments, and they were on track for their growth. So for experiment 2, we have the treatment of halved top and bottom and whole rhizomes with atmospheric pressure plasma jet. And for this particular treatment, we actually treated this in April 5th of 2021. And this was 110 days after the treatment. And these were the average growth heights that we had occurred. So treatment 1, which is the top half treated and the bottom half control, we saw that between the top half treated and the top half control, there was a 8 centimeter difference. And the top half treated actually gave us our highest average growth among all the treatments. For treatment 2, we saw the bottom half treated and the top half control. And between the bottom half treated and the bottom half control, we saw there was a 3 centimeter difference between the two, and that it was significant, but also, it wasn't as significant as treatment 1 we saw. And for treatment 3, we treated the whole treated and the whole control, and we actually saw that the treated rhizome actually produced less growth than the actual control, and there wasn't much of a significant difference between the two. So that's what we gained from that. Here we have figures A and B at the top left. Again, these are June 19th of the turmeric rows after the turmeric had been planted into the ground. As you can see, we have, again, our 4 rows. Figure C at the bottom, we have on June 28th, again, another growth shot of those rhizomes, at those plants as they grew. Again, 4 rows, and yes. Figure D is July 8th, and again, those 4 rows, you can see more growth, and this is pretty much what we did. We just went out to the field, collected those data, and averaged those heights of the 4 replications. So in the conclusions that we had to answer those objectives that we observed during the project, the treatment of rhizomes with both atmospheric pressure plasma jet and plasma activated water was the most efficient in accelerating rhizome sprouting. So those rhizomes that were subjected to both of those treatments actually gained all of the benefits of plasma jet and plasma treated activated water. And for our second objective, we answered that the top treated half of the rhizome yielded the best results in terms of growth, as opposed to its controlled top half rhizome, and overall it sprouted 52.4 centimeters averaged. And for our last point, we wanted to say that atmospheric pressure jet plasma does enable early sprouting and increases plant growth of turmeric. And these are my acknowledgements. I would like to thank Dr. Mantretti, Mr. Lamdong, and Ms. Trang Pham, all of the staff at Alabama A&M that helped to make this project a success. Dr. Shu at the University of Alabama of Huntsville and his team for allowing us to use his plasma lab. And of course, my fellow interns, Kennedy Lipscomb, Alex Drejas, and Michaela Smith. So thank you so much. And a warm thank you to the team at ALREU and CPU2AL for allowing me to contribute to this project. So thank you so much. And these are my references. And this concludes my presentation.
Hi, my name is Flasny, and thank you for watching this video. Make sure to follow us on our social media at CPU2AL on Facebook, LinkedIn, and Instagram. Bye!